Okay, so the recording has started. And before we get into the course content, I want to talk a little bit about the course itself, just to get everybody kind of on the same page. So the first thing I'm going to do is I'm going to um, share my screen so you guys can see what I'm doing. So, all right, can everybody see my Cogbooks page?
Yes.
Okay, so hopefully a lot of the instructions were explained pretty well, but just to make sure, when you open the page, obviously it's going to go to the syllabus, and which is if you scroll down, it'll have it down here. So everybody, be a really good idea, obviously, to read that because there's a lot of useful information about the course, policies, grading, and even the assignments. At the top, you're going to see the most recent announcements, but it's not going to have all of the announcements. So if you think you missed an announcement, you can click on this announcements tab and it will take you to where all the announcements are and you can see them. And you can click on them as they're posted, and then you can read them again, just in case. I like to put a lot of general information to the course so we can kind of, again, keep everybody on the same page. And then probably the most important tab is going to be the modules tab. This is where it's going to get you into everything. And as a lot of you have already discovered, to get started, you have to start with module 0 and you have to go through all of them and get these kind of green check marks in order to open up the rest of the modules. So once you go through all of this, this has really good information. I talked about the course. There's information on me and the TAs in this class. So you'll know which group you're in based on last name, like who's going to grade your exam, so who you can address your immediate questions to about grading. There's also a community forum, which is really nice. I let this be very student to student driven, so people can kind of help each other. If you can't find something or you're not sure on a due date, you can post it there. You can answer other students' questions, and it's where all you guys can kind of communicate to each other. And I will pop on from time to time and make sure there's not any outstanding, questions or potentially wrongly, answered questions to keep us moving forward. Uh, After virtual office hours are completed, I will save the video and I will upload it to right here where it says virtual office hours. If you click on that link, you're going to eventually, as the semester goes on, you're going to have a list of links to the videos. So obviously anybody that can't make it because it's only at, one day at one time, I understand we all have, other expectations and responsibilities outside this course. So we're not going to be able to make all of them, but you can view them all after the fact and use them to kind of study for the exams and hear some important information from the class. All right, and if we go back, there are a couple quick quizzes like your honor lock practice quiz, your syllabus quiz, and your academic integrity agreement. Those you have to take. They're pretty easy to pass. They don't count for any points or anything, but it's good to know that you have read the information, obviously, and then once you take those and finish this, you can open the different modules. And then to get into these, you click on this, and this is where it takes us out of Canvas and it gets us into Cogbooks. And again, as it's been kind of explained in the syllabus, we'll go over it a little bit here briefly. Cogbooks, in a nutshell, it's a series of activities, videos, and readings to replace a traditional textbook lecture-based course. And the benefit is you can do these pretty much at any time. You can do them at any time of the day and you can do them at your own pace. There's a little bit of structure in the sense that the exams are on set 48-hour windows. So is there a question? If anybody has questions, feel free to interrupt and just start with your question. I want to keep this kind of, an open conversation or dialogue for office hours. But back to Cogblocks. So the benefit, like I said, is you can kind of do this roughly at your own pace within the time frame of the exams. Um, I would say a downside to it maybe is we don't have the traditional live lectures. So these office hours, what I'm going to do is I structure them. We're going to go into the Cogbooks materials together, and I'm going to go over them and kind of give my explanation and talk about them in a little bit more detail. So you do it on your own, and then we'll cover it as a group, and we can ask questions during, these office hours. We can talk about some subjects, more in depth than others, depending on where the questions from the group are coming from and maybe what people aren't, are having a harder time understanding. And I do also want to throw out a quick disclaimer. I'm not the course developer, I'm the course instructor. So I didn't make cogbooks or any of the materials associated with them. So we kind of had mixed reviews from students in the past. Some students really like the structure, really like the content and the setup. Some students are kind of in the middle, and some students don't like it. They like, the more traditional, they want, a more of a lecture-based course. But this course is what it is, so we're gonna go with it. And just, as a heads up, I can't change any of the content of the structure, but if you find any errors or you have questions about it, feel free to reach out to me and let me know, and we can kind of work on it together. Um, so a couple students have asked about the format of the exam, which is totally fair. There's going to be the 5 exams. They're all worth 100 points, and they're divided up kind of 50-50. So the first half of the exam is going to be a series of true, false, and multiple choice questions that add up to 50 points. And then the second half of the exam is going to be a series of short answer questions that also add up to 50 points. And the short answer questions are going to vary from one might be worth 8 points, some are worth 10 or 12, and some are worth 15. And the more points they're worth, take into account you're probably going to be expected to answer in more detail, so you'll be expected to have a little bit longer of an answer for higher point value questions. And some of the higher point value questions you really want to pay attention to because they're multiple parts. You might ask, part A, B, and C that are all worth 5 points. You got to make sure that you answer all three of those parts of that question in order to get full credit. Because sometimes it's easy to read, answer the first part, you think you're done and move on, but just go back and make sure you've answered all parts of the question being asked. Um, when it comes to grading, the multiple choice and true false are computer graded. So, those are going to be auto-generated answers, because there's only one correct answer choice. You'll be able to see those the following day after the exam deadline in the next morning. The answers will become available for you to review and see what you got right and what you got wrong with some explanation. Um, if you have any questions about the course content, like in Cogbooks, the readings, the videos, the activities, send those questions to me through an email, the Canvas Messenger, or you can use that question option within Cogbooks. And that's probably going to be the easiest because it takes me to the page where you have questions and I can see the content and the question, and I don't gotta kind of dig up that page, make sure that I'm understanding where you're at.
Question.
Sure.
At the end of the lesson, there's a not sure and a question mark. Use to ask the instructor. Where does that go? Because I've asked a couple of questions and no response.
That goes to my email, and you haven't gotten any responses to those?
No,
Okay, that's good to know because I have replied to a lot of those. Um, and I know that you have sent some, and I did reply to a couple of yours. So I'm going to make a note of that and contact IT because it goes to my email. And then when I reply, I can see my responses. But if you can't, that is problematic.
this
Um,
is in cog books I'm talking about.
yes, yep. It should show up, like you should get a notification in your email that I've commented, and then you should be able to see it in the email or click on the reply and it'll take you to Cogbooks and you should see your question and my response. But if that's not happening, I need to contact IT after office hours and we can take a look at your specific account. Has anybody else in the group that's online also sent me a question and not gotten a reply? Because I thought I got through all of the rest of them today and I was caught up before office hours. Anybody else having that same technical issue? Has anybody gotten a reply from me that's in the group here?
Uh,
I need to go and look and see. I had several questions because there were videos that were not loading or they're no longer accessible, but I haven't
Yes.
seen who replied or not.
Okay, that's frustrating. Not for any of you. That I'm frustrated with the course itself. That if that's a big function that when it works, it's great, and when it doesn't, it's problematic. So, okay, I will reach out to IT and we will get that solved by tomorrow, is hopefully. Because unfortunately, as I'm not a developer, I can't physically do anything to fix that. I need to go through the IT and course developer to see what's going on that end. Okay, we were talking about the exams. Yeah, so we said the first half is true, false, multiple choice. So if you have any questions on the grading of those, send those questions directly to me and not your TA. The TAs are not responsible for the course content or the first half of the exams. For the second half of the exams is where our TAs are going to come into play in our course. Is there are three TAs and myself, so all the exams, all the students are divided into four groups, and we all grade one-fourth of the short answer questions manually. And when you get the grade back on those, you're going to see comments auto-generated from me of what I was looking for, and then additional comments if you lost any points from me or your TAs on where you lost points in the question. And when you have questions about the short answer, do send those directly to your grader. So if you're in group one, the first, quarter of the alphabet, that's my group. You'll send those to me. If you're in the latter part of the alphabet, you'll send those to your TA, and you can find which group you're in on the opening module, the Meet Your Instructor and TAs page. And they should explain where you lost the points from. But when you contact them, unless you say, unless they say something like you're missing part C, and you go, actually, I have part C right here, that should be an easy fix. We're all human, we're going to miss things, occasionally. And myself and they have the authority to give points back if we make an obvious error in grading and miss part of your answer. Um, the other part of that is if you disagree with the provided answer and explanation, please provide a biological explanation as to why you disagree and where from Cogbooks you're getting the information that you disagree with the answer, and we can assess that to give you either full credit back, partial credit back, or no credit back. So there is opportunity to earn credit back in that sense, but it's not guaranteed, and it's really going to depend on your biological argument as to why you disagree. So, are there any questions about the exam format? Okay. And there's a few more comments on exams is they are run through an honor lock system. Some students like it, some students hate it. I don't set the rules on that, as this is how the course is designed. So you have to use honor lock in order to take the exams. It's a third-party proctoring system that do monitor your screen and you through your webcam during the exam in an effort to prevent cheating. Um, Now, obviously, I made an announcement about Honor Lock, but there are going to be instances where you're going to get flagged for doing something. Say, if you get up from your computer to, you're taking it in your bedroom and somebody knocks on your door. I completely understand life happens and we don't live in a bubble. Go answer your door, or you talk to your, significant other, your roommate, or whoever, your child, whoever it is. Um, and that will get flagged, but I will be able to review that and see that you went and answered someone's question and you came back to your exam. You explained, I'm taking an exam, you know, take care of the situation and then go back to your exam. I won't even like you won't even get notified that that'll get flagged. I'll see it, I'll delete it, and life will move on. If I see you looking over your monitor, clearly reading something, behind the screen, that's where, potentially flagging is going to be problematic, where we can have actual evidence of potential cheating. If you have questions about that, you have concerns about it, a lot of students are, I tell them, like, just tell the camera what you're going to do. I understand these exams are for 3 hours, and some students have extended exam taking circumstances that they have, 4 or 5 or 6 hours to take the exam. That's a long time to sit still. If you're getting stiff, tell the camera, I'm getting a little stiff, I'm going to stand up. Just stay in view of the camera. Like a little bit of movement like that is okay. It's just problematic when you walk away from the camera repeatedly. But also, like, we're human, like we're gonna have to use the restroom at times, just tell the camera, like, unfortunately, I have to use the restroom, I'll be right back. It's just part of life. Try to keep it to a minimum. Um, and things will be easier that way. Uh, and also for the exam, you have a 48-hour window to take the exam, but that doesn't mean you have 48 hours to do the exam itself, right? You pick a 3-hour window within that 48-hour window slot to take the exam. And also pay attention. If you wait until 11 p.m. and it's due at 11:59 p.m., you only have 59 minutes to take that exam. You don't get extended time if that time bleeds over past the deadline. So we need to take that into consideration. And everything time-wise is in Arizona time. So I'm currently in Central Standard Time. So I've got to adjust everything on my calendar that everything is an hour earlier in Arizona than it is where I currently am located. Your calendars should auto adjust on Cogbooks, and there is an Arizona a time in Arizona tab that's actually right here, which will tell you the time. But again, it is your responsibility to make sure that you get that exam in because if you take it 2 hours before, when you reach that endpoint, the exam is just going to close, and whatever you have is going to get submitted. So if you don't finish because of that time restriction, unfortunately, that is on you then. Are there any questions about, the exams or cogbooks or the policies like that?
I have a question about if you have a hypothetical experiment, do you have to be species specific like red robin, gray fox, or can you just say A and B?
That's a good question, and those will come up in the short answer questions where you have to write it out. If you're asked to give an example, you can say organism A. If it says give a specific example from cogbooks, the question will be explicit. Generally, I don't like that because I want you guys to understand the concepts more conceptually and big picture than understanding the nitty-gritty details. So I'm not going to ask very species specific questions in the short answer for you to recall. In the or the true false multiple choice, it might say which of these species shows a general negative trend to phenomenon A, and you're going to have to know is it, reptiles, amphibians, fish, or mammals? Like out of those, you'll have to pick. So it's kind of question dependent, but that's a good one.
I do have a question. I guess it's my last question. It's in regards to the supplemental videos and note taking in general. I'm wondering, do we need to take notes on all those extra supplemental videos that we get, such as like the blueberry fields in Maine? Are
No,
those,
the sub. Sorry, go ahead, finish.
yeah, well, go ahead.
Okay, with the supplemental materials, the exam questions were not written using those specifically in mind. The exam questions all come from the main content materials, and the supplemental materials are just that. They're just supplemental to help you understand it in a different way. So like if you're asked to, none of the questions, like a short answer question won't go, recall this scenario in this experiment in if it's a supplemental one. If it's a main text experiment, I may ask you to recall some of the parts of that experiment. But you could go through and not do any of the supplemental materials and be okay. You would have seen all the relevant information. Does that make sense?
Okay, fantastic. That's exactly what I needed to know. Thank you.
Yep. Okay, that's kind of what I have for housekeeping, I'll call it. Now we can get into the actual, why we're here tonight, the office hours. The way I run office hours is I like to go through Cogbooks with you as a group. I do really, really appreciate participation. It makes it go a lot easier. You can use your voice, you can use the chat. But I understand you don't have to answer, every question, and I'm not going to ask. You'll see how it goes. But I do like to get this somewhat interactive to make sure we're, all on the same page. So now I'm not just, talking at you, and you are actually understanding what I'm getting at. So there's a couple questions from the chat, real quick. I want to check. Okay, so we covered this one about the supplemental materials. No. So is there a way to see a list of the content in the course so that we can go back later and review supplemental materials? Uh, sort of. So what they have is if you go into where the modules tab, for each topic, like you're going to have module 1, T1, there's 4 modules for this exam. If you go into the topics and learning objectives at the top, this is going to kind of give you a review and some questions of what we covered and some of the short answer and some of the other questions are very similar to these, and they might expand on it. They might change it a little bit differently. But if you understand this, this is going to be, really show you what you have an understanding on for the topics and what you don't. Does that answer that last question from the chat? Okay, cool. Okay, so real quick, are there any questions about the course like itself, real quick? Okay, if not, that's cool. Let's just go ahead and let's get into the actual Cogblix itself. All right, I stayed open for too long, so I need to. I hate this screen. And obviously, there's a timing out issue, so I'll have to go back in real quick. We'll get there.
While you're doing that, a question.
Sure.
Uh, uh,
Yeah, go ahead with your
Simpsons
question.
and Simpsons and Shannon's indices in the t-test. Would one be smart knowing how to use that for a test, or is that for reference material?
Uh, that's the Shannon index. Um, we'll get to that when we get in here, but those have example calculations. So knowing how to do those indice calculations is going to be important for the test. You're going to see questions that are very similar to the examples and the activities on the exams.
Will the formula be provided, or do we have to memorize it?
Generally, I don't like future exams have a lot of equations, and I allow like a cheat sheet for that. I will have to look at the exam real quick and then get back to you on that because I think they're included on the exam, but I will confirm that later tonight. Is that okay?
Yep.
Okay. We'll definitely go over those formulas and the examples tonight while we're doing this. Um, So getting into Cogbook, so this is module 1, T1. Generally, what they do in the very beginning is they have this think ahead kind of open-ended question. And they don't tell you if you're right or wrong, because the idea is it just wants you to get thinking about the topic in general. So you can write an answer, and you can slide it to whether you understand it or not. And then you move forward. And then if you look down here, if you've already noticed, it goes, thinking back, quantifying biodiversity, and it gives you feedback. So it kind of gives you a question, you read all the materials, watch the videos, do the activities, and then after that, it re-asks that question, and you can kind of get a feedback on that way. They're all set up that way, so it's just kind of a structured thing. So for the first one, it talks about, gives you a video of why diversity matters. And there's 3 really important things that come out of this video. It talks about 3 different types of diversity. Can we recall as a group what those 3 types of diversity are?
Mhm. Ecosystem, genetics, and
Yep.
species.
Yes, exactly. So we have ecosystem diversity, which is just the types of ecosystems. So, for example, it'd be a forest, a swamp, a lake. And then, as you mentioned, there's species diversity, which is the number of species. And then there's genetic diversity, which has to do with the alleles and the genetic diversity of the organisms within that given environment. And then we get into estimating the number of species. And this brings in some mathematical equations. Um, and this is just that. It's an estimate, right? And a big reason why we use this type of practice to estimate it is finding every organism in a single environment is one, really difficult, if not impossible. Sometimes it's really hard to find all of them in there. And two, it would be really invasive and really destructive to that environment. So we want to find less invasive ways to get reasonable estimates on the number of species in an environment, obviously without destroying that environment in the process. So mathematically, let's go over the equation real quick, right? So this is S, that's our estimated number of species. What does S sub OBS mean?
Number of species observed.
Yep, so we have a community that we did like a collection event, and Cogworks didn't really get into a lot of details. Like this could be where you put out a trap for 1 week, right? So you're trapping day and night continuously for 1 week. This could be you walk out and you do a survey for 1 hour and you just write down the animals that you physically, observe in that 1 hour. So you can apply this formula to different types of collection or observation techniques, and it's going to get into it'll
So we
change
were,
slightly. Go ahead.
apologies.
Was that a question or?
No, no, that was my computer going haywire. Sorry.
Oh, okay, no worries. Yeah, so just it's the species that we observe during whatever our observation slash collection slash trapping event happens to be. And then what are these F1 and F2s?
How many were one and how many were two?
Yeah, so F1 is how many species had just one individual of that species observed or collected. And F2 is how many species in that community had two individuals observed or collected, right? And this two, that's just a constant. And then this is the formula, right? So we don't change anything. What we do is we look at our communities. So we went out in this scenario, we went to community A, community B, and community C. And what we found was in community A, we got, 32 wood frogs, 45 green frogs, 12 newts, 15 spotted salamanders, and so on. And we use this information and this equation to estimate how many species are in that community, right? So S observed, that's 12. And that 12 comes from there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 species observed to have at least one organism in community A. So that's where the 12 comes in up here. F1 is how many had a single, right? And that was three. We had isopods that had one, we had diving beetles that had one, and we had dragonflies that had one. So that's three species that had one, and that's where these threes up here come from. The -1 is just a constant in the equation. The 2 is a constant in the equation. But then the F2, how many species had two individuals? That's the ram's horn snail and the amphipods, right? So that F2 is 2. Do the math. 3 times 3 minus 1 over 2 times 2 plus 1 is 12 plus 6 over 1. So it's estimated that in community A, there's 13 species, right? So we've observed 12, but based on this equation, we're going to estimate that we probably missed one. And we do the same thing for community B and the same thing for community C. We'll go to feedback on critical thinking because this will give us the answers to walk through. And we can see how community B and community C mathematically are a little different than community A. A was pretty easy to calculate. For community B, if we use this, we have the same number of species observed, right? Because all of the species had at least one. So that number 12 stays the same. Our F1 is different, right? Because now we have 5 species, the bottom 5, the isopods, the fingernail clams, diving beetles, water boatmen, and dragonflies. So our F1 is 5. And then our F2 What is our F2?
0.
0? Yeah, because we didn't have any species in community 2 that only had 2 individuals. So when we plug it all in, we have our 5 and our 0, we do the math, our estimated number of species in community 2 is 22. And that's, quite a bit more. We had 13 in community A and 22 for community B, yet we observed 12 for each. Why would we have such a higher estimate for B than we would for A theoretically?
Is it something about the estimated number of species in a community increases with an increase in the number of species represented by a single individual? Something to do with that?
Yeah, that's right on track of what I'm getting at. The key is it's rarity, right? So we have a lot of rare species. We had 5 species that only had one in this community compared to we only had 3 in this community. So we would estimate that there would probably be more in community B than community A, because if there's a lot of rare species, it means that there might be other rare species that we potentially missed and didn't even collect anything from. All right, and let's go over the numbers for
Yep.
community C. So, is there a question?
It seems to me that the estimate to me, it seems more like a guess because it depends on how many species were observed, and you don't know how many species you did not observe.
I agree. I mean, in a sense, the estimate is it's like an educated guess, right? Because this method, it's not a complete guess because this method has been validated in different scenarios to show that it is fairly accurate. Um, it's kind of like a calculus equation, right? Where you have derivatives to calculate rate. You're guessing what the rate's going to be in the future, but you can validate it by doing certain types of sampling techniques. And Cogbooks doesn't really get into that. It's kind of, a bigger view picture. So, it's a fair comment, but, this is a standardized, method that we've used in ecology, and it's been validated to be relatively accurate. It's not going to be perfect. It's going to be wrong, at times, but it's going to give us an idea because there's been a lot of patterns and data on rarity and how that influences our estimates on biodiversity.
What is the name of this mathematical model?
Sorry, what was that?
The name of the mathematical model, like what is the name of this equation?
Fair. I'm not.
I actually asked that same question in the community forum, and someone answered that one for me. It
I
was,
think
I
they
can't
had.
even tell you what it was, but it's in the community forum.
Oh, okay.
Usually they have. Yeah, so I don't know the name of the formula offhand, but this is the paper that it stems from. So it was published in Ecology, the Economy of Nature textbook. Um, and this will have a lot more information on the specific equation in this reference.
Oh, are we supposed to have a text?
Supposed to have a what? Sorry, I didn't catch the last part.
Sorry, my mic is not working very well. Are we supposed to have a textbook?
No, there is no textbook, associated or assigned with this course. That's what Cogbooks takes the place of the textbook.
Okay, thank
Um,
you.
for the level of the course, you won't have to know the name of the equation and you won't have to know how it was derived because there is a whole mathematical proof, I'm assuming, associated with the publication of the equation that would be able to explain that. You're welcome to look into that and check on the validation and the accuracy of the model in more detail, but you won't be responsible for knowing any of that information for the class.
I have a question. So should we be extrapolating whether we're going to be using the curve or this equation, depending on the data? So to use this equation, should we be specifically looking for really like diluted populations with high numbers of species counts and then a bunch of low numbers like ones and twos and zeros?
Not necessarily, because because if you get it where your F1 and your F2 are zero, or not, where you don't have any rare species, where your F2 and your F1 are non-existent, essentially, it's just going to be the number of the species that you observed then. Like that's how this equation would work.
Okay, so for the exam, there's going to be a delineation between the curve that's mentioned higher in this on this page, um, as opposed to this equation. Because they kind of, when you're reading it, it kind of bled into each other when they talk about the species accumulation curve. That's a completely different animal when you look at some of the videos that they provide in the extra content. That's a completely different animal than this equation, how they set that
Yes,
up.
and that's a good observation. Yeah, a species accumulation curve, I think, is what you're talking about up here, right?
Yeah.
Yeah, and that's going to be a graph of the number of species observed in relation to the number of individuals sampled.
I think
So
I'm
the
just worried
more
about it being an obvious difference rather than us trying to guess which one we need to use.
you won't have to guess which one you'll need to
All
use
bellies have a trough.
because this is what you would get on the exam is it's going to look like you're going to get a table and
Okay,
you're gonna be asked one potential question could be does community A or community B have a higher estimate of species and why? So then you're gonna have to state, in this case it would be community B has a higher
Listen,
estimate.
listen to this. Listen to this. Excuse me.
Is there a question in there?
No, no, no.
Okay. So I was just saying for the example, for a potential exam question, again, you would get, a table like with data, and it might ask you which community is likely to have more species A or B and why. And we just showed, mathematically, we would say B is more likely to have it because mathematically we can show it in our answer as well as it has more rare species compared to community A. And that would
perfect.
suffice to kind of answer that question. And that's the level that, in this course at least, that it's asking you to derive an answer from this type of data set.
Awesome. That helps me a lot. Thank you.
Yeah, no problem. It's a good question because I know we haven't had our first exam yet, so it's hard to really know what's coming. Let's go over community C still because this is a little different mathematically than A and B, and I just want to make sure that we don't get tripped up later. So for community C, what is going to be our S observed? What's the value of that?
Oh, 12.
8.
Oh,
Okay, so we got one answer of 12 and one answer of 8. Does anybody want to defend their choice?
no, I don't.
8 because we
Okay.
only saw 8 species. There are 4 species where we didn't observe any.
Yep. So that's simply you gotta take that into consideration, right? We did actually observe wood frogs, green frogs, newts, all the way down to isopods because we have, a value greater than equal to or greater than one. We don't have any values where we have values of zero for these last four. So our observed species is only eight. We only physically observe eight in community C. So our S observed would be eight. And then our F1, what value would F1 be for community C?
0.
Yeah, it would be zero, right? Because we don't have
0.
any species where we only observe one. What would our F2 be?
2. 2.
Yeah, two, because these two species right here, right? And then when we plug it all in, we have a zero on top. So species eight plus zero is eight, right? So our estimate is what we actually observed because we didn't really have any rare species. And then if we were to plot some of these, so you might see a plot like this where you have community A and community B. And this gets back to the question when we were asking about a species accumulation curve, right? Because we have
Yeah.
number of species observed here. And on the x-axis, unfortunately, it looks like Cogbooks doesn't have the label in here. The x-axis would be number of individuals sampled, right? So we sample more and more individuals. We generally see a positive trend where we observe more species with more sampling. That makes sense, right? The more we sample, the more we're potentially going to observe. Which community, A or B, do you think has sufficed sampling where we might not need to sample anymore, and
A,
why?
Community A, it's plateauing.
yeah.
Okay, yeah. So somebody said community A that's plateauing, right? So we're getting an increase, an increase, and an increase, and then here these three lines kind of flatline, right? So if we keep sampling, it would probably continue to flatline, versus community B is showing a steady increase. So we need to find where it's plateauing or kind of flatlining in order to suffice sampling. So we probably should continue to sample community B quite a bit more. In community A, we probably have enough to suffice our estimate is fairly accurate. Are there any questions about how to calculate the estimated species observed? Again, if some people are going to be at different points. If you haven't gotten to the material that we're going over in office hours, that's great. You can ask questions later. If you don't, if you find you have questions later, shoot me an email or send them through the website itself. Okay, so here we're talking about patterns of abundance. And really, kind of, again, in a nutshell, what this is looking at is you have your abundance category. So it's going to be low abundance to high abundance. And then the number of, this is a bird species, this is number of plant species. And what they want to kind of really point out here is that most species, so your highest number of species are going to have kind of a medium abundance. You're going to have a low number of species that are going to be found to be rare, and you're going to have a low number of species that are going to be found to be extremely abundant in a given environment. Most species are generally kind of at an intermediate level of abundance. Okay, so then this is talking about kind of some of our rank abundance curves, and they give us some figures here, right? So rank abundance is going to be showing simply that you're going to have less species here and more species here, and you're comparing two different communities. And you can get a few different pieces of information here, right? We know that species evenness is one thing on here. If you have a flat line like this, you're going to have a very even environment. If you have a kind of a trending line, you're going to have an uneven environment where these are going to be your relative abundance is going to be high and your relative abundance is going to be lower as you go down here. So you have an uneven community for community A and a relatively even community for community B. But it also tells us species richness. How does this type of plot, a rank abundance plot, tell us anything about species richness? What do the actual dots on the plot tell us? What do they represent?
Do they represent each individual species?
Yeah, so they represent a species, right? So there's the richness in this community here in A, well, in this environment, I should say, in graph A, is the same, right? Because they each have 5 species, right? So the richness itself is equal. Over here, the richness is different because in eucalyptus plantation, there's only 5 dots, so 5 species, versus the primary forest, there's several, right? So there's a lot more rich in the primary forest than it is in the eucalyptus plantation.
So is it safe to say that on the graph, the steeper the slope, the less evenness there is in a community?
Yes, that's a good way to look at it.
So I had a quick question on this. I know these can go back into a normal bell curve for it because it's right now ranked in order of, which one is most abundant to which one's least abundant. How would I know which side of the bell curve they would fall on?
Uh, I'm not sure I fully follow how these would go back into a bell curve. You're asking,
Yeah, could I put it back into a bell curve? Because it said that it's their log means
so
it's a
you
bell
look
curve
at,
and then it
are
goes
you talking about like
into
these curves?
Yeah, how would I put it back into that from there?
So this is log transformed data.
All right.
So they used log transformation data, so it's not actually to rank it.
Mm-hmm.
In this plot, you wouldn't necessarily, you wouldn't, I mean, one, you wouldn't need to do any sort of
Okay.
log transformations. It doesn't get into that math side of the equation. It just wants you to be able to, you see the plot, how do you interpret the
Okay.
data? That's the important part here.
All right. Okay. Thought I
Fair
get that.
question,
Thank
though.
you.
On the math note, are we going to be using limits or anything like that? Calculations to find populations or anything? Because I noticed that
Uh,
in comic books they talked about the curve that we were talking about earlier, and they had mentioned finding the asymptote, and I kind of stopped right there because I wanted to ask how deep into the math we needed to go.
no, you won't have to find the asymptote of those curves or anything like that.
Okay.
And as far as the math goes, we'll get into an example in a little bit. But if you see in the activities and the readings where there's like an equation, and then there are examples on how to do the math itself, and there's some practice problems with it, you'll likely have to repeat that and do that math and that level of calculation on the exam. But there's some equations in here where they just introduce it and they don't give any data or any practice with it, and you won't have to do any of the calculations with those types of equations. For both, you'll probably have to understand some of the theoretical. It might say, like, if this variable increases, the estimate also increases, true or false, right? Like, it won't be a calculate this, but it'll be a theoretical question like that. Does that make
Okay.
sense?
Yeah, no, that's really valuable. Thank you.
Okay, no problem. Okay, so now we get into calculating diversity of species. And we have these two indexes, right? The Simpson's index and the Shannon's index. And they both, again, we have a data table, we have a formula, and we have an example. So it would be fair game on an exam to give you a data table and ask you to do the calculation. And the way the
Okay.
Simpsons index works is you just have to be able to essentially pick out where the values come from, right? So we have a 1 over the summation of P sub I, or the relative abundance squared. And the way that looks is because it's a summation, it's all of the P sub i's. So this is where the 0.24, the 0.16, the 0.08, those are all coming from this column right here, the P sub I of community A. And then we can repeat it for community B, and we can repeat it for community C. And then when we calculate it, we're going to get a value. And based on which index you use, that value is going to tell you if your diversity is high or low. So for the Simpsons index here, right, the lowest value you can get is 1, where there's no diversity, and it can increase. So obviously in this example, it's 4, so it's, that's greater than 1. There is some level of diversity in that environment. It's not completely one species. And that makes sense based on uh, the absolute abundance that we observed in these environments.
I asked before, but for clarification, the formula will be provided. All we have to do is put the data in.
Yeah, I'm just gonna answer that now and make a decision. I forget exactly what's on the exam, but I will double check and make sure the formulas for these will be on the exam.
Thank you.
Yeah. Because I'm not a big fan of memorizing formulas. I'd rather have you learn how to do it and learn the material. Because you can always reference a book to get the formula, so it's not like it's hidden from us in the real world.
Amen.
Okay. And then this one is the same way, right? We just have a different formula, the way they arrange it. And the minimum value is 0, which represents a community that contains only one species, so no diversity. And then the maximum value, just the natural log of the species in the community. So this is more important understanding that if it's above 0, you have some level of diversity and how to actually plug these in. And one thing I kind of forgot earlier about the exams is I did get emails about calculators. Really, the only restriction is you can't use your phone as a calculator, but you can use a basic calculator. You can use a calculator on your computer because during the exam, the uh, the proctor is going to be able to see what's on your screen. So if you pull a calculator up on your screen, they're going to see it. You can use scientific calculators like the TI 8491 or whatever. I don't even know what number they're up to now. You can use those. So really, the biggest restriction is no tablets, no and no phones. So
I have an Inspire. Are we allowed to use that?
I'm not sure what that is. Can you tell me what that is, quick? If not, we can talk about that offline too, which might be easier if you wanted to send me a follow-up email. Um, Basically, the big thing is we don't want to have something that has internet connection. So if you use your phone, you can either be calculating or you could be Googling, the answer. So
Apologies, I have one more question. This is in relation to the Shannon's index. I was looking at one of the supplemental videos, and it showed a completely different way to calculate or a different formula for the Shannon index. And it was a little bit confusing because when I tried to use the version in the supplemental video, it didn't come out to what the answer was for this version of the formula. Does that make sense?
it does. I'll be honest, I'm not positive which supplemental video you're referring to. I will look into that. But for the exam sake, use the formulas on this page.
Got it. Okay.
Like that's the method that is intended. But I'll make a note of you said
I'll
it's for
post
the Shannon or the Simpsons index?
this, Shannon. I'll post it in the chat real quick.
Okay, thank you. Okay. And then the rest of this section is kind of just that critical thinking, what determines biodiversity, and then getting some feedback. If I have this right. Let's come up. I don't personally really like these questions because the content doesn't really get into anything about light, water, oxygen, and heat and abiotic factors. So, for the sake of the exam, the way I formatted it, you can, I mean, take these into consideration, but you're not going to be tested on these questions or this content. Same for the second one. Competition, mutualism, and predation. Those are all units that we're going to cover in later modules, not associated with this exam. So don't worry about knowing what competition, mutualism, and predation are. You can basically ignore this page. Um, I don't know why they format it this way, so I'm not, holding you responsible for it. These would be better questions later in the course, in my opinion. Yeah, so are there any questions then from module one? So that pretty much covers um, everything. And again, if questions come up later, feel free, please reach out to me. I like getting student questions because honestly, sometimes I bring stuff up from questions from students into office hours because I know if one or two students aren't understanding something, that's going to mean there's more than that. Um, so please don't ever hesitate to ask questions. But if there are no more questions on module one currently, we can go ahead and go to module two, or technically module one T2. They changed the structure of this a little while back. Okay. So then the first thing they get into is they talk about landscape ecology and really got into habitat heterogeneity. So if we're looking at this picture right here, would we say that there's a lot or a little of habitat heterogeneity, and why? What
I
do we
would think
see in
it's
the
a
picture?
lot because there's water, there's a higher elevation area, there's hillsides, kind of islands.
Yeah, very good. I totally agree, right? We see all sorts of different types of habitats in this picture. As described, we see waterways, we see forests, we see potentially grasslands or hillsides, we see other dark grass or vegetation over here. So yeah, we see a lot of differences, which by definition means we see a fair amount or a lot of heterogeneity. And then based on the title, it kind of gives us the answer here that heterogeneity enhances diversity. But I'll ask, why does heterogeneity enhance diversity, or how does it enhance diversity?
More niches.
Yeah, more niches or more resources, right? Animals need vegetation to eat potentially, but they also need water to drink. So they have, forest can be their shelter, grassland can be their feeding place. So if you have all, different resources available, you can create more niches and have more diversity in a given environment. And this just really, what this example kind of shows is that kind of the legacy effect that was introduced on the previous page, where people lived here and they brought resources to that environment, and having multiple resources in a given environment actually had an effect on the number of species. So as they got further away from that settlement, it got less heterogeneous in the environment. So you had lower species richness the further you got away from this settlement. So it's kind of just a cool kind of like real world example of it in action. And then this gets into quantifying biodiversity, and not in the mathematical sense, but more in the theoretical kind of definition sense, right? Because we have local diversity or alpha diversity, which is like going to be more small scale diversity. So this might be something like a single pond or a single grassland, kind of field, or a single field, maybe I should say, versus you have larger scale where you have regional and gamma diversity that might be like kind of the whole wetland area or the entire forest itself. Or maybe even like you could do it like on a city scale where you have you define it just on a larger scale that's going to generally have more diversity or more heterogeneity. And then it gets into beta diversity. What is beta diversity? Or when do we use beta diversity?
It ecologists refer to the number of species at different occurrence between two habitats.
Yep. So by definition, we got that. And when we use it, it's just when we're comparing habitats. So if we have habitat A and habitat B, and we look at, a difference in number of species, the beta diversity is if habitat A has 10 species, habitat B has 4, the difference between 10 and 4 is 6, right? So that's beta diversity, pretty simplified.
That's the difference, you said, correct?
Yes, it's the difference between the difference in the number of species between two communities. Okay. So then the next section gets into talking about larger areas hold more species. And what these figures are showing is just island area, right? So we have zero, so small islands to larger island area, and we get this positive trend of the number of species. Why do we think that island area has an effect on the number of species?
More biodiversity.
Yeah, right. Just like exactly what we've been talking about. You have more area, so you have more potential for more biodiversity or more heterogeneity in that environment. You have more resources available in that environment. And another thing is you also have more space. So if you have more space and more resources, you also have generally less competition. So you can then focus your energy away from competition with other species to reproduction and have more species and more offspring within a species even. Yeah, and then this is just another example using kind of a log scale. And then this is what I was getting at here is they bring in species area curve, this formula, but there's no examples and there's no practice, so you won't be expected to do any type of calculations with this. The example I was giving is true or false, as A increases, S decreases. What would we say to that? As A increases, does S decrease?
No.
No, it would be false, right? Because as area increases, the number of species in the area also increases. So that would be the type of question you could get, expect to see from these types of formulas. And this is just another real example of, land area and the number of species using birds. Same pattern. And this breaks it down even further by different species, right? We have plants, amphibians, and reptiles, birds, and mammals. They all show this positive trend. As we increase land area, we increase the number of species. Okay. Then it gets into habitat fragmentation. Does anybody want to kind of describe what we mean by habitat fragmentation?
It's where habitats are, um, they're not contiguous, so they're surrounded by a different type of habitat.
Yeah, right. So it breaks up that continuous habitat. So in this example, we have the green color is going to represent, I think rainforest, and then the yellow color is going to represent where that rainforest is kind of broken up. And why does habitat fragmentation occur? What's one of the biggest reasons that it occurs?
Land or human development.
Yeah.
Yeah,
Yeah.
humans in a nutshell, right? Humans develop it for agriculture. So they cut the forest down to make fields. They cut the forest down as a product, right? So they can use it as timber. They develop cities, and it just really fragments these different kind of habitats. What are some non-human human causes of habitat fragmentation?
Flooding, fires.
Yeah, so more natural disasters, right? Flooding, fires, we said, could be hurricanes, could be disease, could potentially come in and fragment habitats of like birds or mammals or amphibians, the habitat itself, parts of the habitat. So very good.
First question, do trees count as part of the habitat, or is it more just like abiotic?
So the habitat can be defined in a couple different ways. You can define a habitat just by land area, and then by default, everything, birds, insects, plants, water would be part of that habitat. Or you could define the habitat by like species if you wanted and say like we're looking, but it's usually not by species. You can say like, this forest habitat, and within the forest it contains trees, birds, insects, even like fish in the ponds in there. So it's a little subjective, but you can make it objective by explicitly defining what you mean by a given habitat. It could change based on what you're looking at. Does that make sense?
It does. I was just thinking because, when there are, for example, palm forests are planted, it's a forest, but it's not like the natural habitat. It's very, very different monocrop, doesn't have biodiversity, etc. So Like,
It would
but
still
it's still
be, it could still be a
trees.
monocrop habitat. Like you could
Yeah.
say this square mile is the habitat, that's how I'm defining the habitat and everything, the part what makes up the habitat is every living thing and even non-living things. Like part of the habitat could be soil, it could be rocks, a
Mhm.
mountain could be part of the habitat. So it doesn't have to be necessarily biotic things.
Okay.
Okay, so then this one talks about how fragments influence biodiversity. Um, what did we get out of this? Does fragmentation increase biodiversity or does it decrease biodiversity?
Decrease.
Yeah, it decreases it. What are some of the ways and why it decreases it? So we think of it in terms of like human development, and we have a forest, and we're defining our habitat as a forest. If we fragment that forest with human development, we're actually just taking away that natural habitat. We're reducing the area of that usable habitat. Um, It also increases edges, right? And they talk about edge effects in this a little bit. Why are edges not generally considered great for biodiversity?
And that have something to do with like fluctuation in temperature, like it could be drier or hotter on the edges of that area, which some, species can't handle that temperature fluctuation, like less shade or too much, um, something along those lines.
Yeah, you're right on track again. It directly affects the abiotic conditions such as temperature and humidity because it gets rid of that interior core of that habitat. So it's exposed to the outside elements at a higher level. And they gave some examples also that, you know, this species right here of cowbird is a habitat specialist. So you might lose like 10 species to edge effect and gain one or two. Your net effect is still -9 or -8. But why are these species particularly bad for or the environment? What is their impact or what do they do?
They're parasitic.
Yeah, they're a parasitic bird, right? So these cowbirds they don't make nests. The females rather will go lay their eggs in other species bird nests, and they what they do is the host nestling is usually smaller and develops slower, so the invasive or parasitic bird here will outcompete the host nest, and the parent will kind of blindly feed that invasive cowbird and therefore it lowers its fitness because it's raising another species offspring and it's losing its offspring because there's even evidence and example or documented cases where these cowbirds will actually as nestlings push out the the host nest or push out the host nest nestling or push out the egg. Um, they're actually pretty cool videos showing that. So they do a really good job of reducing the reproductive output of the host species. And they're attracted to edges, and they do particularly well in that environment. So having more edges will attract and increase their numbers, which will decrease the numbers of the other species. Okay. And then we get into how biodiversity, it like with a lot of ecology, it's not always black and white. There's, there's always that it depends kind of factor. And one of the things that biodiversity largely depends on is colonization and extinction rates. And this gets at, some things to consider is they use islands as an example. And you have the mainland where the source population comes from, and then an island off of that mainland where it gets colonized by that source population. It doesn't always have to be an island. This could be ponds. moving from one pond to another pond. It could be moving from one patch of forest to another patch of forest, one field to another field. So it's useful to think of it, or easy to think of it in terms of islands, but islands don't always have to be land masses and water. Um, and some things that matter are the size of that island, so the area, as well as how close or how far those islands are from the mainland population. And what it'll get into is this is just some examples of, when you have larger area is going to have more bird species. We kind of already talked about that. And islands that are larger and nearer to the mainland are going to have even more bird species. So they kind of break it down and get a little more technical with it. And that gets us into this theory, right? The equilibrium theory of biodiversity. And what you have is you have kind of a multi-part figure. On the x-axis, you have number of species on the island, or in the environment, we could call it, but we'll just stick with island to stay consistent with the text. So you have 0 on the left side, or low numbers, and high numbers as you go to the right. And then on the left y-axis, you have rate of colonization, and we see a negative trend, right? So rate of colonization goes down as the number of species on the island increases. Why do we see that negative trend? Why doesn't it stay flat, or why isn't it positive?
Because you run out of resources eventually.
Right, right.
Well, that's with extinction. It's not necessarily that you run out of resources. Say you have a mainland with 10 species, and on the island you have 0, and then you all of a sudden you have 1, 2 that colonize it. But once it gets to full colonization, like 10 out of 10, you can't recolonize it once you're there. It's not migration, it's colonization. So there's a difference there. So as more and more species get there, there's less to get there. So that's why the rate of colonization goes down. And then you have on the other y-axis, you have rate of extinction. So as the number of species on the island goes up, you have a positive trend where the rate of extinction. And because you have more species on that island, you have more possibilities of extinction. So that's why with more species, more can go extinct. And this gets a little bit into the competition of when you have more species, you generally have more individuals and you use up more resources. And that's why rates of extinction can increase when you have more species on there, because this is where you're getting at using up some of those resources, or at least using up that space. Okay, and then they show that it's not, it's even a little more complicated, right? Because then they talk about large and small islands. These trends tend to, when you have a larger island, you have a lower rate of extinction compared to when you have a smaller island. And that gets at what we've talked about a few times already, right? Larger islands tend to have more space, so less competition, and they have more resources. So your rate of extinction, or your likelihood of extinction, is going to go down. Versus on a smaller island, you have less space, more competition, and less available resources, so you're more likely to go extinct, which will give you a higher rate of extinction. What are these dots? Like we had one up here, and we have 2 here. So what are these dots representing?
Intersection between colonization and extinction.
Yeah, so where colonization and extinction are equal, right? And that kind of gives us almost like a point of equilibrium. So the extinction rate and the colonization rate, when they're equal, it's going to be balanced. So we're not, the populations are going to be fairly stable at this intersection. And it's not stable, the stability changes based on size, right? So we have this point right here. The number of species is at this level on a small island, but when we have a bigger island with more resources and more space, that number of species on the island moves to the right and it increases, right? So we can support more species, or a greater number of species, on a larger island than we can on a smaller island.
Does that equilibrium mean that thing like it's stable, so nothing is happening in either direction, or
It
um,
doesn't,
the
sorry, go
rates
ahead,
are
finish.
happening at the same time, like the colonization and extinction are happening simultaneously, or it just has stabled out? I'm a little confused on that.
Yeah, so it kind of both. So it does mean that colonization and extinction rates are equal, when they're equal, you'll find stability. So like the colonization rates and the extinction rates, they can change, right? If a storm comes in or a disease comes in or a fire happens, you're going to get a higher rate of extinction, right? It's going to move on that the extinction axis. But they kind of sway, back and forth a little bit. You'll get a bunch of organisms that'll come in and over colonize, and then when we over colonize, there's going to be die-offs because they can't support it for too long. If we have die-offs, it's going to swing to the left, and we're going to get opportunity for more colonization to move it back to the right. So it's always going to kind of like a pendulum where it's going to sway left and right slightly, but it's always going to kind of try to stabilize or at least come back to this point. Does that make sense?
Yes, thanks.
Okay, no problem. Okay, so that's talking about the size of the island and how that has an effect. But as we mentioned earlier too, there's also a distance portion, which is going to affect colonization. So if it's far, you're going to have a lower rate of colonization versus if the island is near the mainland, you're going to have a higher rate of colonization. And why do we see the difference here? Why is there a higher rate of colonization for islands that are near or closer to the mainland?
Greater ability for a higher variety of species to be able to reach it.
Yeah, so I think part of it just comes down to species of locomotion ability, right? If you have small organisms that fly but can only fly a short distance, they might make it if the island is really near to the mainland. But if that island's miles away from the mainland, they might not be able to make it there. Some organisms might not, they might be able to swim a short distance from the mainland to the island, but if it's way out there, they just don't have the stamina or locomotion or locomotive ability to make it out there. So it's just more challenging for them to physically get there. There's more probability of them getting picked off by a predator on the way. So again, just there's several reasons why it's more challenging to get to a far island than a near island. And it does the same thing, right? So it changes that point of equilibrium. A far island is going to result in lower numbers of species than a near island. And then in here, they kind of put it all together, right? So we have four possible points of equilibrium in this scenario based on if it's, small and far, that's going to have the smallest number of species that it can support. If it's large and near, that's going to have the greatest number of species that it's going to be able to support. And then kind of your small and near and your large and far intermediates. And this is, a very simplified version, if you will, of this theory. There are many other variables that you can add to this and grow on this and do individual environments. But this is kind of just like, a big, general view of some of the major factors that affect colonization and extinction as far as being able to support the certain number of species on an island.
For the exam, should we expect some of the scenarios to be more granular as we're trying to negotiate these questions, or will it be more general, like what's in the book?
It's definitely not going to go much really like it can't go beyond what the materials in the cogbooks are. So focus on what's there. You might get some scenarios like true or false. A small, large island can support more species than a small, near island. So
Okay.
it could be that simple, or it could be a short answer question where it says use the theory of island biogeography to describe the patterns that distance and size has on biodiversity. And then you're going to have to write, 4 to 8 sentences describing all of these different patterns.
Okay, thank you. Because there's just like such an infinite number of variables that could, make distance look a lot larger if it's like really windy or if it's, high seas or something like that. So I just wanted to make sure we weren't going to go that far into it.
Yeah, any the additional variables like that I talked about, even like predation levels, like those are you can put those in there, but if they're not covered in cogbooks, you're not going to be held accountable for it.
Okay, thank you.
Yep, no problem.
This seems too intuitive.
It's meant to be, to a certain level. Like you can take it further than this, but this is the level that they want to use for this course. And I mean, I'm glad. I mean, being intuitive is a good thing, right? Because these
Yeah.
are general trends that we do see. You saw previously, this is very clean, right? Because this is
Yeah.
all theoretical. If we go back to some of the figures, they're a little bit messier, right? Like this is not as smooth, but it's a general trend. And the same thing with this. Like not all the data points, are the same. There's some differences even within, like the same area. They have different number of species, but the trend is there. And that's where ecology
Mm-hmm.
rarely is really clean, where you get, like a perfect fit line. It's a lot more trends because there are a lot of unaccounted for variables. If we modeled every variable possible in that environment, we'd have models that would be super complex that are trying to predict reality to like the second, and that's just not feasible.
Mm-hmm.
And animals don't. It's hard with human studies. You can ask somebody questions and they can answer you, right? You can actually talk to your study organism and get answers. With animals, you can't really talk to them, and sometimes they do things like that are really random or seemingly random. So that's why the data are a bit messier. Okay. And then they talk briefly about how corridors promote biodiversity. So, how would we describe what a corridor is?
It defragments the habitat.
Mm-hmm.
Yeah, kind of. Maybe a better way to put it is it connects different, it connects similar habitats, right? Because right here we have this bridge that's connecting two forested habitats. The bridge itself is not forest, right? But the bridge is a structure that animals or organisms can use to move from one habitat to another in a safer fashion, right? Because the point of the bridge is to go over the highway and avoid the traffic rather than go across the highway. So, there's definitely the benefit of, that's the intended benefit is it increases the safety of the organisms. But can anybody think of some negatives possibly of a corridor, at least like, this example in this picture, why a corridor might not be always the, a positive thing?
Is it because it forces species that wouldn't otherwise be together to interact?
Yeah. So what it can do is it can kind of condense a lot of species or a lot of organisms to a small space. And if you condense a lot of organisms and make them interact in close quarters, you might have something like bacterial transmission, potential disease transmission. There's going to probably be a lot of defecation in this area, right? So it's going to make, passing parasites possible. Um, and it also, what are predators going to do when they learn the use of the corridor?
Mm-hmm.
They're
Mm-hmm.
going to learn that the deer are going to tend to cross here maybe, and wolves are going to be like, oh, we just got to sit here. We don't have to hunt them all in this entire forest. We can just hunt them on these two edges. So it can kind of really bottleneck your prey species to be right at your predators. Because some predators are pretty smart. So what they do is they usually take that into consideration. You'll have multiple corridors down this highway to kind of mitigate some of those problems. So it's not a perfect solution. It addresses some issues. From a human perspective, it's nice to have corridors too because, you don't want to hit, moose or deer or other animals with your vehicle. So it prevents, human fatalities or car crashes. But they do need to consider the effectiveness. Like if you only have funding for one corridor, it might not be worth it to fund that single corridor until you can build multiple up there.
We here in Utah, we have a corridor over one of our major highways by the mountains. And even with that corridor, we have a whole herd of elk that have been running up and down the highway or the freeway actually for the past couple of days.
Sure. I mean, and if a disturbance happens at that corridor, like all of a sudden they run into wolves on the other side, they're gonna learn like, oh, they're over there, we're gonna avoid it. So they might learn to avoid corridors. So like it really depends on, the species and the interactions and how effective and useful they're gonna be.
Yeah, we have one herd that pretty much almost annually uses our freeway system.
That's crazy because that's on one hand it's really cool probably to see, but on the other hand it's extremely dangerous, I
So
imagine, and
so much.
terrifying. So okay. And then it gets into designing kind of a preserve after we talk about the physical shape of the landscape. So what it has on this activity, we can do it together real quick and talk about the answers if we want. So on the left, we're going to have a worse or bad design, and on the right, we're going to have a better or good design. And this is talking about reserve size. So if this is our good or our better design, what would be a worse design?
Smaller.
So a smaller one, so like this one right here, we're thinking.
Yep.
Mhm.
Why is a smaller reserve a worse design than a big reserve?
Less biodiversity.
Yeah, it's a pretty simple question, right? You're going to get more biodiversity as we saw with more area because we're going to have more resources and more space. So if you're going to make a nature reserve, you want to have more biodiversity and more space so you can protect more organisms. B, they did it for us. So the number of reserves, they're saying one big one is better than four small ones. And why is that?
It creates those patches, edgings, matrix things that they have to cross, that forces animals to, some animals can't cross that that matrix and it kind of forces them into that zone.
Yeah, so
Less
say that
fragmentation.
absolutely. Fragmentation is
Fragmentation.
one. Moving through the matrix is one. Say that these four patches represent four different parts of the environment, right? We have a pond, we have a forest, we have a field, and we have maybe swamp or something, right? So if the organisms need to use those in this reserve, they're going to have to exit each one, and we don't know what's between this. This might be, a road that's really dangerous. It might be a river. Um, it might be like a field that makes a prey really vulnerable to cross from one patch to the other patch. So it's better to have it, a continuous reserve rather than have it broken up. Okay, and then this one, C is talking about reserve proximity. So it's got
All
four
right.
uh, patches that are close together, and then a worse design, I would agree. It would be bottom right. It would just be moving those patches in proximity as distance, right? So if they're further away, they have to exit the reserve over a longer distance and for probably a longer period of time, exposing them to more danger potentially versus moving within these. It's a lot closer together. It's also an energetics thing, right? It's going to be more energetically expensive to move across these four patches than it is to move across these four patches. All right, what about D? Reserve proximity again.
Second from left. Second from left.
So you're saying this one?
Yep.
So the proximity would be the distance, like that would be the same, right? Because they're the same distance away from each other.
It's
So
the
the
one off.
bottom right.
Yeah.
Yeah, so
That,
what they're getting at is the bottom right,
Mm.
where all four of these patches, the
Hmm.
distance between them is closer than say the front one and the back one. Like these are really far apart
Yep.
compared to any of these. And you could make some arguments for multiple answers. This is kind of which is the best answer and why. So
All right.
reserve connectivity.
The
I go
middle.
with second to left this time again.
Yeah, exactly. It would be this one, right? Because these are patches that are not connected, and these are patches that are all connected. So connectivity, and then reserve shape. We've got two left. Which is the better to
Our,
design, an oval or a
our
circle?
left on the left. On the
So we
right.
got
That one on the right. The one on the
this
right
one.
to the right.
Okay. So we got
Yep.
a circle would be a better design than an oval. I agree. Why?
the oval has edges, right?
Yeah, there's going to be more edge on the oval compared to the circle, and this has more of the interior core compared to here. So like the distance across it is going to be less than it is going to be across this one. Yeah, so we can double check, submit that. Folks, do your thing. Yep. All right. Green check marks across the board. So we got those. And we talked about why, which is important because you're not going to get a matching like section on your exam, but you might get a compare and contrast these different nature preserves, design a nature preserve, and explain four variables of what you would consider and why each variable is considered and which one is better than the other. So those are the types of questions that you might get related to this activity. And that pretty much covers all the materials because the rest of this is just some videos to watch about, we talked about this already. Islands aren't always surrounded by water. They talk about different types of habitats. They're, good videos to watch. So I recommend doing that. Does anybody have any questions about the T2 module here that we didn't get to yet? And if not, that's totally fine. I mean, we covered a fair amount of ground. We're gonna have another office hour tomorrow where we'll cover the materials in T3 and T4. And then you'll have your exam one next week ready. So keep that in mind. This is a very fast-paced moving course. But yeah, if there's no more questions, we can call it, a session. And if other questions come up, we can definitely you can definitely, send me a message and we can kind of communicate that way. I'll say I do appreciate, those that were able to make it and participate. To your classmates that will watch this later, will definitely, I think, appreciate that as well.
Yeah, thanks for clarifying so much.
Thank
No
you so
problem.
much.
All right. Well, then I will see maybe some of you tomorrow. Have a good evening.
Good night.
Thank you.
Yep.